Thanks a lot for having me here. It's lovely to be back in Paris. So I'm going to talk about fossil traces of brain evolution. And here you can see one of my main focus points. So this is a hominin fossil. So this is a fossilized skull and a brain imprint of an Australopithecus from South Africa that's at least, say, 2 million years old. And so I work at the Max Planck Institute for Evolutionary Anthropology, as Jean-Jacques just mentioned. And my colleagues and I find different ways of trying to answer the question, what makes us human? And I myself and my research group, we study the hard evidence of human evolution, the fossil remains. And we mostly work with microcomputer tomographic data or synchrotron data, as I will show you. And the approach we take is we compare our own species and variation in modern humans with our direct ancestors and our closest relatives. In this case, you can see a modern human skull next to a Neanderthal skull. And I think even if you're not a paleoanthropologist, I think it's easy to appreciate that they don't look the same. And so what makes modern humans different is from Neanderthals is a much smaller gracile face and a round brain case. And the round brain case can tell us something about what was inside once. So even in fossils, we can use these endocranial imprints that are created in the course of development inside the skull and make informed inferences about brain size and brain structure from these endocranial imprints. And the comparative approach that we're taking is not only comparing to fossils, but is also comparing to our closest living relatives. And basically, the main theme of my research is trying to understand why evolutionary changes of developmental timing have such big effects. And here you can see how similar a bonobo and a modern human fetus look like before birth. And as they keep growing and developing, you can see in this rendering how the pronounced differences that you see between the adults emerge in the course of development. And basically, the core, and this is why I'm here today, differences between these closely related species are in the developmental timing, and they're mediated by differences in gene expression. And the classic framework, theoretical framework to describe these differences in evolutionary timing is called heterochrony. And I think it's important to mention that the term was initially coined by Ernst Haeckel, the famous or infamous biologist from Germany. And this is one of the drawings that he used to illustrate his biogenetic law. We now know that this is not correct. I mean, the law that he postulated that the ontogeny, so the development recapitulates phylogeny, is not exactly correct. And he was forcing it a little bit by manipulating, so some would say, his drawings. But basically, this is where the term heterochrony came from, as an exception to what he thought was a biogenetic law in biology. Later on, this term took on a different meaning, and I would say that the most influential use of that term comes from Stephen Jay Gould's work in the 1970s. And this famous Harvard professor basically tried to use that model of heterochrony, and he developed this influential clock model. And what you can see here is that basically he envisioned changes in shape and size. And what's important, I think, in this model, and it's also true for what came after Stephen Jay Gould, is that the changes that are being envisioned are changes that regulate the entire organism, right? And this inspired, I would say, the search for a master regulator or several master regulators of development. And in this heterochrony view, modern humans basically retain childlike features of our ancestors at much larger size, right? So here you can, on the left hand, see, again, these are still images from the animation that I showed before. So basically modern humans have, in this view, like a baby face, but they're bigger, and they have a rounded head, but bigger. And I think what I want to challenge today is the idea that there is one single master regulator, one single clock. What I want to convince you of, that even when we're talking just about the skull, we're looking actually at evolutionary changes to many different clocks that are not perfectly synchronized. So they are coordinated because the organism and parts of the organism need to grow and work and function together, but they're not perfectly synchronized. So my main topic today is going to be brain development. And I think it will be no surprise to you when we say that when we compare a chimpanzee brain scan to a modern human brain scan, that the biggest difference is that the modern human brain is more than 3 times larger than those of our closest living relatives. There are also differences in the organization that, and they're most pronounced, I would say, in the inferior frontal region, including Broca's area that is responsible or is a key part of the human language network, and also in the back of the skull. So what we know is that brain growth and brain maturation takes longer in humans than in chimpanzees. Okay, and so one of the human characteristics is that we have a long childhood, and that has been linked to brain plasticity, and the brain plasticity is thought to be a key feature of our cognitive abilities. So the big question for today is when and how did this particular mode of brain development evolve? And we're looking at fossil evidence in a comparative framework, as I mentioned before, and we're going back several million years of hominin evolution. And basically, we phrase this question as how ape-like in the brain development was Lucy. So Lucy, as many of you will know, is a famous representative of a hominin species that lived in East Africa more than 3 million years ago. So the formal species name is called Australopithecus afarensis. And they have a key position in the hominin tree because they're likely ancestral to all later hominins, including ourselves, right? So basically, we studied them, because there are many beautiful fossils, not just Lucy, available, and we studied them because they give us kind of a baseline for basically the hominin condition. And the reason why we not just compare them to humans, but also to great apes, is because you can see how ape-like, at least in the overall cranial morphology, these skulls of Australopithecus afarensis were. Right. So on the left hand, you see a CT scan of a chimpanzee, and on the right-hand side, you see a reconstruction of a female adult Australopithecus afarensis. And the reason why the chimpanzee has a cut here is because this is part of an ongoing collaborative project where we study not just the skull, but also study the brain of the same animals. And these are from zoo populations and from populations in the wild. So when an animal dies of natural reasons, then our veterinarians go in and directly extract the brain, even if that site is in the middle of the jungle in the Ivory Coast. And they preserve the brain in formaldehyde, and then we ship it to Leipzig and MRI scan it. And the CT scans of the skull and the brain allow us to basically study the endocast and the brain of the same animal. Right? So we can basically ground truth the patterns that we see on the endocranial imprint inside the skull and connect this to different modalities of the brain. And you can see here on the right-hand side that we also do something called diffusion tensor imaging, where we can visualize the white matter connections in the brain, not just the outside shape of the brain. And I'm happy to report that every major groove leaves an impression inside the skull. So this is really important because that allows us to directly connect what we see on these endocranial imprints to the underlying brain morphology. So when we talk about development, we need juvenile individuals, or ideally babies. So for Australopithecus afarensis, we have two beautifully preserved infants. And one of them I will talk about in detail today, because this is the Australopithecus afarensis child from Dikika in Ethiopia. And this was found by my colleague Seraya Lemsiget, who is now a professor at the University of Chicago. And he and his Ethiopian team found, now 2 decades ago, I think, found a partial skeleton, and they brought it to Grenoble to have it synchrotron scanned. And this is what this synchrotron scan of Dikika looks like. This is a partial view. And you can see basically what must have happened is that after death, the cranium filled with some fine-grained sediment and turned into stone. So we do have a natural impression of the inside of the skull. So my postdoc, Simon Neubauer, and I spent literally years of our lives manually segmenting those synchrotron scans. So we basically labeled the different parts of the bone and the endocranium. And then the fruit of this labor you can see here. So once you remove the fossilized matrix, you can peek inside the skull, and you can see that even the most delicate bones on the orbital roof, they are perfectly preserved. The problem is that because of pressure in the fossilization process, they're not all in the exactly correct place. So we had to take the skull apart and then put it back together, and that's I would say several years of my life, where we basically tried to reconstruct the original shape of this individual over and over and over again. Right? So we don't just use one reconstruction, we use many reconstructions to basically also account for the uncertainty of the reconstruction process. And what you can see appearing here in white is the natural endocast of this individual. And then we were able to compare the brain imprint of the Dikika child to similarly aged chimpanzee MRI scans. And we were able to basically map many features that we can observe on chimpanzee brains and endocasts to the Dikika child. And we could conclusively identify the sulcal imprints. What we found is that it basically has an ape-like brain organization, so it doesn't have some of the derived features that your brain and my brain has, especially not in the in this inferior frontal sulcus, so it doesn't have a reorganized Broca's area, for example. So what we also wanted to know is how old exactly was this individual when it died. And my colleagues, Paul Tafro and Adeline Le Cabec, were able to basically use synchrotron images of the dentition to count the daily growth lines that you can see in the teeth, which is really amazing. So basically, a tooth grows in daily increments. And because you grow at different speeds during the day and during the night, you have basically daily growth lines, and then you can count them like the rings on a tree to determine how old that individual was when he died. And you connect different teeth in the upper and lower jaw by these stress events that you can also recognize on these synchrotron scans. So counting this, they were able to determine that this individual died when he was 861 days old. Okay? So this is around 2.4 years. And of course, there's an error on that, but the area of margin is really, really small. So it's basically, 2 weeks in either direction, right? So we know really well how old this individual was. Another thing we need if we want to reconstruct the ancestral brain growth pattern of Australopithecus afarensis is we want to know how big were Australopithecus afarensis babies. Okay, unfortunately, we do not have neonates or fetal remains of this species, but what we do have are beautifully preserved pelvic remains. Right, and here you can see, for example, the comparison between the pelvic shape and the birth canal of a bonobo female. And we also micro-CT scanned bonobo neonates. And on the right-hand side, you see Lucy's pelvis. So Lucy is thought to be a female Australopithecus afarensis. So it's basically the potential mother of this Tikika child. Right? It's not the literal mother because they are also 300,000 years apart, but you get my point. And if we basically simulate using this bonobo neonate, how big the head can be, so it would fit through Lucy's pelvis, we can basically constrain the neonatal head size and therefore have an estimate of endocranial volume at the time of birth. So now we have almost all the pieces of the puzzle. We have adults, we have children. There's another child from Hadar, that's the same site in Ethiopia where Lucy is coming from. That's basically dentally the same age. It's died when he was, around 2 years old. And then we can compare these individuals in these larger comparative frameworks where we can look at modern human brain growth patterns, or endocranial volume patterns, I should say, and chimpanzee patterns. And you can see, okay, two things are strikingly obvious. So modern humans grow their brains differently than chimpanzees. So far, no surprise, right? So they grow fast in the beginning, and they grow for a longer period of time. And this is how they end up being more than 3 times as large than chimpanzees. And in absolute volumes, these Australopithecines, they look very chimpanzee-like, right? I think that's pretty clear. So the 1 and 2 labels here are the 2 children that are died shortly after their 2nd birthday. And the adults are a bit larger, about 20% larger on average than chimpanzee adults. There's another way to compare these growth curves. So we can look at the relative endocranial volume. We can just standardize the adult volume to 100%, and this makes it easier to compare the human and the chimpanzee growth pattern and see where the Australopithecines fall. And what's, I think, striking are 2 things. A, there is some overlap, but it's clear that basically modern humans grow over a longer period of time. This is why their relative growth curve in blue is underneath the chimpanzee curve. And no matter how you reconstruct the chimpanzee endocranial, the Australopithecus endocranial volumes and which individual pairs you compare, the Australopithecus afarensis, they're basically lower than the chimpanzee individuals of the same age, which means they basically have a more human-like, protracted pattern of brain development. So what we've argued is that the first detectable step of hominin brain evolution is involved a longer period of brain growth, right? And this is based on the direct comparison to chimpanzees. In absolute terms, they're really very ape-like still, but in relative terms, they grow over a longer period of time. And that's important because that would imply a longer period of brain plasticity that already emerged more than 3 million years ago. And in humans and in other apes, we know that this brain plasticity has largely to do with the establishment of the white matter networks inside the brain. Which parts of the brain are connected and how myelinated they are, right? Because at the time of birth, you already have all the neurons that you will have as an adult, but the neural circuitry is still sparse. So this is really what's driving this postnatal volume in all primates. What's also true in all primates is that if you have a protracted brain growth that is correlated with a longer dependence on caregivers in the widest sense. In primates, that's usually the mother. In humans, it's usually a larger group. This is why in the publication we were careful to say it's just caregivers, because we don't know if they're dependent on the mother only. So we think, okay, that this growth and maturation rates may already give us a hint about changing of social behavior and dependence. But the question is this really something that we can say just based on the comparison between humans and chimpanzees? Because as soon as we broaden the comparative perspective by including not just chimpanzees but also other great apes, the picture gets a bit more complicated, right? So if we include gorillas, orangutans, and chimpanzees in this picture, we can document considerable diversity in these brain growth patterns among extant great apes, right? So there seems to be a continuum of variation among great apes, and not just a dichotomy or that all great apes grow the same compared to humans. So I think we need to be a bit more careful. And what we are suggesting is that basically these systems are highly flexible, right? That brain growth patterns can vary even among closely related hominids, and there's no reason to assume that they cannot vary among hominins, right? And there's other evidence that Jean-Jacques Hublin published some time ago, where it's not clear, for example, how protracted and prolonged the brain growth in Homo erectus was, for example, right? So our suggestion is that maybe this is also something that's evolving within the hominin lineage and not just something that evolved once and was there to stay. And I think it's important to emphasize that basically the dichotomy chimp-like versus human-like is not enough to capture the complexity of hominids and probably also not of hominins. So we are also arguing basically that this prolonged pattern of brain growth in Australopithecus afarensis is not a mere byproduct of brain size increase, because that is basically what you see in every textbook, is that the reason why we grow for such a long period of time outside the mother's womb has to do with the evolution of upright walking. Okay? And this is called the obstetric dilemma hypothesis, because if you basically redraw a gorilla to stand as if he were a modern human, then you can see that among all the things that are changing, pelvic shape is also changing, right? And basically, compared to our closest living relatives, modern humans have an extremely narrow birth canal, which is often considered to be an adaptation to upright walking, but that of course makes it more complicated to give birth to a large-brained baby. So what we are basically arguing is that, okay, we think these projected brain growth patterns in Australopithecus afarensis maybe set the stage for these changes in social behavior, but they may not have been the initial driver. These changes in the social structure may have come later, maybe much later. And our alternative interpretation of the evidence, and that largely comes from the direct comparison with the orangutans, who basically in these patterns that I showed you before are the closest match for the Australopithecus afarensis brain growth pattern. And in orangutans, it has been argued that basically this long protracted brain growth pattern and also the long dependence of the infants on the mother, they are not weaned until they are 8 years of age in the wild, is actually a strategy to distribute the energy needs of the growing brain over a longer period of time. And so we are arguing that maybe the initial driver of these changes in the brain growth pattern in hominins was an energetic trade-off rather than a social change to begin with. And with that, I'm happy to thank all my collaborators and co-authors. I want to thank you for your attention, and I'm happy to take any questions you may have. Thank you.